Welcome to the channel Bharat Pharmacy. So in this video, I'm going to explain about the starch hydrolysis, one of the biochemical tests which is widely used for the identification of bacteria to confirm whether it is secreting any enzyme called amylase. So starch hydrolysis is one of the unique biochemical tests to confirm the ability of the given organism to produce hydrolytic extracellular enzyme, an enzyme which is synthesized and it will be released into the environment. Amylase is capable of degrading the polysaccharide starch. So for doing this experiment, the requirements are 24-hour-old culture of Bacillus subtilis, one of the positive organisms which is producing the extracellular enzyme called alpha amylase, and media starch agar, which is rich of starch source, and reagent iodine or Gram's iodine. So what is the principle behind in this starch hydrolysis? Starch is a high molecular weight branching polymer composed of glucose molecules are joined together by glycosidic bonds. So because of the high molecular weight, this glucose molecule or the starch, it cannot enter into the cell wall. So by the extracellular enzyme, so whenever the organism is realized the outer environment it is containing and starch, it is able to secrete an extracellular enzyme called amylase. This amylase is an adaptive enzyme. So there are two types of enzyme called adaptive and constitutive enzymes. Adaptive enzyme is the one it will secrete by the bacteria or the organism only when there is a specific substrate is available. So whenever the bacteria is introduced into the starch containing medium, the bacteria is able to secrete and this amylase. In the absence of starch, bacteria, it won't synthesize any amylase. And later, this macromolecule of starch is converted into shorter polysaccharides, mainly dextrine and maltose, and finally into smaller glucose unit. The final hydrolysis of maltose yields glucose that can be transported into the cell and used for the energy production through the process of glycolysis. And starch is a complex polysaccharide. It consists of two components, mainly of amylose and amylopectin, so which are present in various amount. The amylose consists of D-glucose units linked in the linear fashion by alpha 1,4 linkages. And the amylopectin is a branched polysaccharide and it's a shorter chain of glucose units linked by alpha 1,4. And this is also joined to each other by alpha 1,6 linkages. And this starch can be hydrolyzed by an alpha amylase, which is secreted by only few genera of bacteria. So from this amylase secretion, so we can confirm some genera to certain extent. The ability to hydrolyze starch is used for the identification of most of the organism. And the positive result, when you are introduced into the starch containing medium, if the organism is able to grow, so those places the starch is completely utilized. And by the time when you pour Gram's iodine on the starch agar medium after 24 hours or 48 hours, there is a blue coloration. And if clear zone of hydrolysis around the organism growth, that indicates that the starch is hydrolyzed. And if there is no hydrolysis, that indicates that the organism is not able to synthesize the extracellular enzyme is called alpha amylase. So most of the bacteria are able to produce extracellular enzyme. And since the starch is too large molecule based on the molecular weight, it cannot diffuse into the cell membrane. And that is the reason the starch, it has to be broken down into smaller units. So the organism is having ability to secrete an adaptive enzyme called alpha amylase. Only those organisms is able to degrade the starch into smaller units of starch into glucose. And whether this hydrolysis, whether the starch is degraded or not, so that we are confirming by pouring iodine in a solution or Gram's iodine, and based on the color production, either of a clear zone of hydrolysis or dark blue color with the Gram's iodine, we can confirm the production of alpha amylase enzyme on the starch agar medium. And generally, this is the overall principle. A starch containing medium, if you add organism, this is able to secrete and alpha amylase, it acts on the starch molecule and it produces a dextrine. And later, this dextrine is getting converted into maltose. And there is another enzyme called maltase. It acts on this maltose and it converts into smaller glucose molecule. And these glucose molecule, it can enter easily into the cell wall small because it's a smaller molecule. And if the organism is able to produce the alpha amylase enzyme wherever it is growing, around the growth, you can see a clear zone of hydrolysis where I have mentioned is positive for Starr-Zucker test, where those organism is not able to secrete an alpha amylase, you cannot find any clear zone of hydrolysis around the growth of organism. So from this way, you can easily confirm whether the organism is able to secrete the alpha amylase enzyme on the Starr-Zucker containing medium. So all the genera of bacteria doesn't have the ability to secrete the enzyme alpha amylase. Only few genera is able to secrete the alpha amylase. So based on the zone of hydrolysis, you can confirm whether the given test organism is able to secrete the alpha amylase or not. Thank you. We'll meet you on one more video with some other topic. Thank you.